Hello everyone, Gauri here. I hope you all are doing well. And in this session, we will be exploring the gram positive and gram negative bacteria and its staining technique, which is one of the most important staining techniques in microbiology. The gram staining technique is named after the Danish bacteriologist Hans Christian Gram, who developed it in 1882. Gram staining is basically the primary step for the initial identification and classification of the bacteria into two groups, that is, gram positive and gram negative. It stains the bacterial cell wall differently, giving gram positive bacteria a violet color and gram negative bacteria a pink color. As a result, bacteria's cell walls absorb color, which improves their visibility under a light microscope. Additionally, the analysis of the morphology and arrangement of the bacteria is aided by gram staining, and based on the differences in cell walls, it also enables us to understand the chemical and physical properties of gram positive and gram negative bacteria. During the procedure, the bacteria are first stained with crystal violet and next treated with iodine to promote dye retention. When gram positive bacteria then are treated with ethanol, the alcohol is thought to shrink the pores of the thick peptidoglycan. Thus, the diiodine complex is retained during this short decolorization step and the bacteria remain purple. In contrast, gram negative peptidoglycan is very thin, not as highly cross linked, and has larger pores. Alcohol treatment also may extract enough lipid from the gram negative outer membrane to increase its porosity further. And for these reasons, alcohol more readily removes the purple crystal violet iodine complex from gram negative bacteria. Thus, gram negative bacteria are then easily stained red or pink by the counter stained safranin. Although several explanations have been given for the gram stain reaction results, it seems likely that the difference between gram positive and gram negative bacteria is due to the physical nature of their cell walls. And true structural difference between the two groups of bacteria did not become clear until the advent of the transmission electron microscope. The gram-positive cell wall consists of a single 20 to 80 nanometer thick homogeneous layer of peptidoglycan or murine, which is lying outside the plasma membrane. And in contrast, the gram-negative cell wall is quite complex. It has a 2 to 7 nanometer peptidoglycan layer covered by a 7 to 8 nanometer thick outer membrane. Because of the thicker peptidoglycan layer, the walls of gram-positive cells are more resistant to osmotic pressure than those of gram-negative bacteria. These are some of the examples of gram-positive and gram-negative bacteria. I hope you enjoyed watching the video. Thank you.